Hello, Year 9. Welcome to lesson on the immune system. So we're getting our way towards the end of this topic. We've looked at so far how our bodies are able to defend ourselves from preventing pathogens from getting into the body, and now we're looking at what happens if they do get their way through those chemical and physical barriers. So we're looking at the immune system today. Same as always, we've got our starter here in front of us. So please give yourselves about 6 or so minutes. Answer as many of these as you can, and then we'll go through the answers in just a second. So pause it when you want to start and unpause it when you're ready. Okay, so question 1. Which barrier to infection covers the body? It is, of course, the skin. The skin covers your body. Why does that barrier protect against infection? It does because it is thick and it is also difficult for the pathogens to get through. There's very few openings in it. How are trapped pathogens moved out through the lungs? So they're moved along by the cilia from those ciliated cells. How does the chemical defense in the stomach help protect against infection? So the chemical defense in the stomach, if you remember, is hydrochloric acid, which will kill many pathogens that get through. Which chemical defense against infection is found in the tears and the saliva? It's an enzyme called lysozyme. How does this chemical defense protect against infection? It breaks down bacterial cell walls. So chlamydia and HIV, how are they both transmitted? They're STIs, sexually transmitted infections, and they can also be transmitted, as you remember, from mother to child while in pregnancy, and also through the sharing of needles, so blood barriers. Remember, one way that the spread of explain one way that the spread of chlamydia or HIV can be reduced or prevented. Any kind of prevention of contact with sexual fluids, and also breast milk to babies. What usually triggers the immune system response? Any pathogen inside the body will trigger that response very quickly. And what name is given to the molecules released into the blood by the immune system cells? You may or may not know this. We don't think we've covered it yet. Antibodies. Well done. Right, let's move on. I'll leave you to go through these at your own pace, so pause it if you want to stop and unpause it when you're ready. Right, we have got a lot of keywords today. There's a lot here that you will know. Something like a vaccine you'll almost certainly have heard of. Herd immunity, if you've listened to the news at any point in the last 3 months, you'll have almost certainly heard people talking about herd immunity. But there might be other words here like antigen or lymphocytes that you may not have heard of before. So please do, as always, make notes of these keywords, ideally in your own words, but obviously you can do it word for word if you like. Pause it when you're ready and unpause it when you're ready to move on. Right, so a couple of definitions to go forwards. These will make a lot of sense as we move into the lesson, but it's really good. It really does help if we know what they are before we talk about what they do. So we start off with our two gens, G-E-N words. The pathogen we know is something that causes the disease, and the antigen is something that covers the outside of it. If you can find a fun way to remember those, please do let me know. I don't, I'm afraid. Obviously a little question down there in the corner for you. If you want to have a little think about before we move on to the next bit, but that diagram, I don't know if we come back to it. I think we do later on, but if we don't, it might be worth making a note of it now because it covers quite a few different aspects that we are going to be looking at in today's lesson. So when you're ready, obviously pause and unpause as always. So the last definition we need is the antibody definition. So these are these little Y-shaped things that we saw a minute ago. These are proteins that cover white blood cells. So they are on the outside of white blood cells. White blood cells we'll now start calling lymphocytes. That's their scientific name. And it's our body's way of tackling pathogens. So the immune system will use these antigens, will use the antibodies to attack the antigens. That's where things get really fun. So, a little task for you. Read the statements on the sides, look at the diagram on the other side, See if you can match the statements to the keywords. Don't worry, we will be coming back to this. It's purely just a guessing game at the moment. Probably best on a whiteboard or scrap paper, whichever suits you best, to be honest. Pause it when you're ready, unpause. Those are the answers, but I don't think we need those just yet. Right, the immune system. So I will talk you through what's happening here. Some of the animations work, some of them don't. I have given up trying to make them work. They're annoying me too much, so I do apologise. So we have the lymphocytes and we have the pathogens. Lymphocytes are good, pathogens are bad. Our body uses lymphocytes, there are white blood cells, remember, to attack pathogens. Nasty, nasty pathogens. So on the outside of the pathogens, you should be able to see, let's see if this is going to work for me. You should be able to see these lovely arrow-shaped things here. These are the antigens. These are the defining features on the outside of a pathogen. Each pathogen has its own unique identifier and is identified using its antigens. On the outside of the lymphocyte, we have these called the antibodies. Now these are attached to the outside and they are detachable. These will be key in just a bit. Right, let's move on. So step one, pathogens have antigens on their surface. This makes them unique. So as you can see on the outside of the pathogen, we've got the little arrows. These are the unique antigens to that pathogen. On the lymphocyte, on the white blood cell, on the left-hand side, you see you've got those Y shapes. Those are its antibodies. You will notice these fit a bit like a lock and a key. That is because that is what they are designed to do. Very similar to enzymes, as we'll talk about in a bit. So when the antigen meets one of the pathogens, it becomes activated. So this means that the antibody that's relevant on the outside of that particular white blood cell can become activated. Once activated, it triggers this initial stage of the immune response, so responding to the pathogen. So once the lymphocyte is activated, we move on to step 3, and this causes that particular lymphocyte to reproduce itself over and over and over and over and over and over and over again, very, very, very rapidly. This is why infected areas of the body can become warm. It's partly due to trying to combat the infection, but also increasing the temperature helps speed up this metabolic rate. It's also a bit of a byproduct from it. So now we have lots of lymphocytes with lots of antibodies on the outside of them, and all they've got to do is find something to attack. So what they'll do is they will release the antibodies from the outside of their kind of cell membranes, and they will race towards the pathogen. So you managed to get that one working. They will race towards the pathogen and they'll attack the outside of it. They'll attach themselves to the outside of it and force the cell to undergo lysis or some other process that will destroy it. One thing to bear in mind is that once a lymphocyte has secreted its antibodies, once it's used its antibodies, it can no longer copy itself. So that's why it copies itself before releasing the antibodies, so that it can release as many antibodies as possible. And finally, once the infection has been fought off, so we can assume this little guy here is now destroyed. Once that has been fought off, there will be some lymphocytes that will remain in the blood, the ones that didn't fully activate, and they will sit there and they will wait, and they are called memory lymphocytes. This means that if that same pathogen comes back, rather than having to wait to be activated and copy itself, these are there waiting, ready to go. They can rapidly copy themselves and attack it, probably before you've even noticed any symptoms of the infection. So that's the very basics of our immune system. There is a lot more to it. I've kept it about as basic as I can. If you want to go into any more detail, there are some amazing YouTube videos. All you got to do is Google or YouTube, how does the human immune system work? And there are some insane videos out there. So please do, if you're interested, have a read up of some of those. So moving on. Oh yes, sorry, I forgot to put that there. So this is the step-by-step. Now you can either go back, skip back through the last couple of minutes and use my description to create your own flow diagram, or you can use this particular flow diagram here. It doesn't matter which one you use, so long as you have got a a description of how your body responds to a pathogen using antibodies and antigens. This first meeting, as you can see up here in the green box, is called the primary response. Primary school is your first school, primary colours are the first colours, the primary response is the first response. This is when you'll feel often most ill from an infection, because the pathogen has had a chance to multiply and move its way through the body unchecked while the immune system responds. And so often you do get more ill the first time you suffer a particular infection. Lovely little fact there in the red box for you as well. Right, when you are ready, pause it to make your notes and unpause it when you would like to move on. So, immunities, building up an immunity. Immunity doesn't mean that the pathogen can't get into your body or the pathogen doesn't infect you. It does. What it means is that you're able to respond much, much faster to the point where your body can fight the infection before any perceived symptoms occur. This is because your body has memory lymphocytes. Please make a note of that. They're the antibodies that match a specific pathogen's antigens from a primary response. They, as you can see there, make the secondary response much faster. You've got that lovely little graph there. The way to think about it is that when the curve is at its peak at any point, it is fighting the infection, and when it is at a trough, there is a no infection. So you can see the primary response, it takes about 4.5 days to start to fight the infection effectively, and then you can see there that it doesn't start to slope down until day 18. So it takes 8 full days for your body to fight off the infection. Then, after some period of time, your body gets infected again. You can see that within 4 days, your body has built up enough antibodies to fight off the infection. The slope begins to drop down again, and your body has fought off the infection within 5 days of receiving it. So near enough half the time, which is brilliant. It means you don't suffer any of the side effects, or not side effects, symptoms of that particular disease, and you are considered immune. Make your notes, then move on. So, a lovely diagram there for you. If you want to sketch it into your book, I would strongly recommend it. But please do have a shot at both these questions. The answer will follow. Questions into your book, please. Pause and unpause when you're ready to move on. So, the answers are as follows. The secondary response is faster because more antibodies are produced. It's about the speed of that production. And then, as you can see there, the more rapid production, the more rapid production of larger antibodies in the secondary response will kill it before you're even knowing you are ill. So this actually is quite important now that we're moving into the track and trace stage of fighting the coronavirus. It means that you may, or somebody in your family may have suffered the infection previously, but it doesn't mean they can't still carry it. They will be able to fight it off much, much quicker, because in theory they will have some sort of immunity, or at least some sort of memory lymphocytes in action, but they are still contagious if they have been in contact, which is why the track and trace system is so important. Right, unpause it, pause, unpause, whatever, when you're ready. Right, vaccinations, very, very quickly. We're not going to go into the controversies of vaccinations in this particular lesson. I'll leave that for another day. Because that's a nice little debate to have that's very difficult to do when you're listening to a video. But vaccines contain inactive or weakened pathogens. Sometimes they contain alternative pathogens, so a pathogen that it has very similar, if not identical, antigens, but is not the same pathogen. And they are injected most of the time. They can be ingested, they can be eaten into the body, and it will cause your body to have an immune response. Your body's immune response will then be able to trigger the memory lymphocytes to remain in the blood, and you are now immune to that particular disease. We tend to do it as a child because children don't want to be getting these infections, and so the earlier you can vaccinate safely, the much better it is. Now, with some vaccinations, there are some side effects that can come because if you are injecting a weakened pathogen into the body, that is still a live pathogen or an active pathogen, and so can still sometimes cause particular symptoms to occur, such as fever or nausea. So there are some symptoms that can happen. However, these are significantly lessened compared to the actual infection itself, and are normally easily dealt with a bit of Calpol. Right, once you've made your notes on this slide, please pause, unpause, whatever, move on. Right, can you please arrange these? And once you've finished, have a shot of that extension. What is your definition for immunity and vaccine? Pause it, unpause it, we'll go through the answers. So at number 1, I hear you shout, it is of course a person is injected with the weakened or dead version of the microbe. Then yes, you are correct, the white blood cells produce the antibodies to fight off the microbe. And third, oh, you got it, if they get infected, in the future the body will recognize the microbe and antibodies are produced much quicker, as we saw in that graph previously and as I described. And finally, if you can destroy the microbe quickly, you are not going to suffer any symptoms, or at least you will be suffering significantly less symptoms. Right, let's move on. Right, our final check for you today. Into your books, can you please answer as best you can, and obviously in full sentences and all the rest, blah, 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 blah. Explain why being given a vaccine for measles makes you immune to measles. Pause it and then unpause it to get the answer. The answer is, of course, that the vaccination causes the body to make these memory lymphocytes, these antigens to the measles pathogen in the vaccine. The memory lymphocytes will recognize measles in the future and cause a very rapid response to stop you becoming ill. And with measles, it can also significantly reduce how infectious and contagious you are during those early stages. And finally, a practice exam question for you. And once we have answered this, I will say goodbye to you, and you've got your worksheet that attaches to this lesson that you can have a shot at. Pause and pause when you're ready. And your questions are there for you to see. Right, I will have to say goodbye to you right now, and please do have a shot of the worksheet. Have a go at the quiz that's going to crop out for the lessons this week, and I will see you next lesson. Toodaloo!